All right. DNA to proteins. This is literally going to be a chapter on just genetics. And we're going to take a look at this one piece at a time. And we're going to start off with the scientific experiment that broke this whole thing open and started us thinking about what DNA was and how it worked. And that was the Robert Griffith experiment. So, Robert Griffith loved to kill mice. That's one of his favorite things to do. And he was looking at two forms of something called pneumonia. Pneumonia is a disease that slowly fills your lungs up with fluid, making it hard for you to breathe. And if you're a mouse, well, there's no hospital for mice, so that's that. If he gave you the S bacteria, the mouse died. Okay, so the S bacteria, the mouse died. And if you gave you the R bacteria, the mouse lived. So pneumonia S killed you, pneumonia R, and you lived. Then if you took the S, you took the vial of the S and he cooked it. He literally heated it. Uh, that heat would kill the bacteria, and if you would inject the heated S version into a mouse, the mouse would live. So here we have regular S kills you, R you live, heat killed S you live. So we grabbed these two, right? The R where you live and the heat killed S where you live, right? And the mouse would live in both situations, and he combined them into one shot. And what he found was that these mice would die. And he's like, well, why is it if I grab R that doesn't kill you and S that doesn't kill you, why is it that that would combine to kill you? So, different ideas started coming across, but what he settled on was something was being transferred, something was being shared between these two types of bacteria, and whatever was being transferred uh, uh, is what allowed this combination to be killed. But there wasn't a lot of certainty as to what it was that was doing this. Then came Avery. So Avery was like, "All right, let's see if we can't figure out what is being transferred." And what he looked at was literally samples of the thing that was being transferred. He was able to isolate it, and when he isolated the transferring chemical, he looked at the chemical composition. So he looked at the percent of nitrogen, the percent of phosphorus, and the ratio of nitrogen to phosphorus, right? And you don't have to memorize this table, but he's literally just looking at these numbers and seeing how they're related to each other. He's like, okay, 14, 15, 9, 15, 3, 13, 4. All right, 8.5, 9, 9, 8.5. Okay. 1.6, 1.7, 1.6, 1.5. All right. He just looked at the numbers and then he compared them to the numbers he knew for DNA. And what he found was these numbers were similar. These numbers were all similar to this number. And these numbers were all similar to this number. So this suggested that DNA could very well be the thing that's being transformed from one version of the pneumonia to the other. And notice I said could. And that's the issue here. We did not eliminate all other possible options, so they weren't done. Then came Hershey Chase, and Hershey Chase was looking at something called the bacteriophage. A bacteriophage is a virus that infects bacteria. And the way bacterial infections work, nothing goes into. So when this virus infects a bacteria, nothing goes into the cell except for the virus's DNA. So the only thing that goes in is the virus's DNA. And what they were trying to do was to show that what was being transformed from one bacteria to the other was in fact DNA and not protein or anything else. Like it was just DNA. So what they did was they radioactively labeled DNA with radioactive phosphorus. This is phosphorus that would emit radioactivity that could be measured using a machine called the Geiger counter. It goes click, 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 really fast when you get really close to something that's radioactive and not when you're further away. And then they also did a separate experiment where they used radioactive sulfur, which would be in proteins. There's no sulfur in DNA. So they used radioactive sulfur And that sulfur would be in proteins to see if any of that went into the cell. And what they found was the protein stayed outside, but the DNA is actually what went inside. And with that combination, they knew from the Hershey Chase experiment what was being transformed was actually DNA and not something else. And that's it for chapter 8, section 1.